So I will talk about transient rewiring in vision. And basically, what I want to propose to you today is a new paradigm to think on how the brain works and how the mind emerges in brain, how the visual perception emerges, how do we see the world. And to introduce this paradigm, I have put this beautiful drawing by Ramon Kayal of a pyramidal cell to remind us how we typically think about neurons, what has neurons, what we have told to ourselves as neuroscientists, how the neuron works, what is the key transition between neuron activity and perception, and then to tell you the new approach that I'm proposing. Right? So traditionally, everybody knows, I don't need to explain it. We think as the critical thing about neuron is the output, the activity, the firing spikes, the firing rate, maybe the electric potential across the membrane. That's what's relevant for perception, right? So if we perceive something, that's what neurons do. They perceive it through neural activity, right? Through spiking activity. And now you may be surprised that I will tell you that I'll try now to convince you to start thinking in a different way. Right? And to introduce that, think like this. This is the classical paradigm where we have fixed connections. And we have lots of inputs in the neuron. They get summed up in some way, there's output generated. If there was nothing else than this in a neuron, then of course, there's nothing else. So we have to base all our explanations of how perception emerges from neural activity based on activity outputs of neurons. However, there's this other possibility, right? That neurons are able to transiently select some pathways. So switch some pathways on, switch some pathways off. In this case, for example, in this example, we have the one in the black is selected, the ones in the white are not selected. And this could change 100 milliseconds later, 200 milliseconds later could change. The neuron could switch to another pathway and say, okay, now I let these inputs in and do not let these outputs in. And there are presumably some smart switches, or you can call them gates, that close and open these pathways. It's not inhibition. It's very different from inhibition. The effect may look similar at the beginning, but it's not the same thing. And the reason that it's not the same thing is that these switches last much longer. Inhibition will last typically 20 milliseconds. After 20 milliseconds, any inhibitory effect will wash out. These things last several hundred milliseconds, for example, right? So what are these magic doors, magic gates, right? So we have on the membranes, we have proteins that are called G-protein-gated ion channels, which actually can change the properties of the membrane and can keep their states for much longer periods of time. And we also have metabotropic receptors that can activate those ion channels. And the metabotropic receptors respond to ligands. So that's the basic mechanism that I'm proposing. Now, how could this work? Like here's one example where you have a person driving a car. And let's say this person has decided to drive a car and it has to form a feedback loop for controlling the steering wheel and keeping the car in the lane. And so this has to involve, of course, spiking activity of the neurons, but it has to form a specific feedback loop that exists only for this task. Later, when the person is doing something else, cooking at home, it doesn't need this feedback loop. It needs some other feedback loop, right? So this is how it would work. So we would have a feedback loop that is formed by particular selection of particular pathways, and that eventually end up with the states of the G-protein-gated ion channel. Of course, in this short talk, I cannot explain lots of details how this would work and what the implications are, but I invite you to read this paper. There is a lot more information in this paper that was published last year. But what I do want to explain is and what the implications are. How do we have to, if you decide to give a chance to this idea, to this hypothesis, how you have to start thinking differently, how you have to start interpreting your data differently. And any data in neuroscience that you look at, including the results we have just seen in the previous two talks, one can start thinking differently, how we could interpret the same results with this new type of paradigm. So here's the difference. Here's how you have to think differently. Classically, the result of cognition, we always consider the output of a network. So you have here input on the network and the output of the network. Now, in the new paradigm, when we still have inputs and outputs of the network, but we also have the new transient states of the connections that last for several hundred milliseconds, then change again, and so on. Now, these states of the networks are now the result of cognition. There's also output from neurons, but outputs create behavior from one side. They also feed back into these connections, and they provide the neurotransmitters for the metabotropic receptors to make new changes in the connections, of course. But the state is the state of our mind is the state of these connections, right? So this is what we think. So, for example, like the classical example in neuroscience, grandmother. Where is the grandmother? Like when we perceive a grandmother. So you have either grandmother neurons, or you have a population of neurons that fire for a grandmother. Now, what I'm suggesting here is that we have grandmother metabotropic receptors and grandmother G-protein gated ion channels. When these ion channels are open and closed, and they set up a particular network, subnetwork, rewire the system for grandmothers, for processing grandmothers, perceiving grandmothers, responding to grandmothers, and so on. Right? So that's a very different way of thinking how the brain potential could work, and requires quite reorganizing our thinking. Right. So to show you how different this is, I want to also mention. Let us think what kind of adaptive mechanisms we have had traditionally in neuroscience, and what I'm proposing to change, right? So traditionally, we have basically two levels of adaptive mechanisms. We have a network that produces cognition and behavior, both of it. And then we have another adaptive mechanism, which is plasticity mechanisms, which train the network. We change the network. Right, so we have these two things. One is slower, one is faster. That's how neuroscience works traditionally. Right? You can think of them as two big boxes. Now, by introducing this new concept of this intermediate levels, so we have metabotropic receptors are something that in G-protein ion channels, it's something that works much slower than the network. It's much slower than the spiking activity, than inhibition, excitation, and so on. It's hundreds of milliseconds and not tens of milliseconds. And yet it's at the same time much slower than plasticity, right? So it can be thought of as a third adaptive mechanism that is somehow sitting in between the classical two, right? And this may have been the missing box, the missing part to explain how cognition emerges from neural activity, or how perception emerges from neural activity, right? So the third box, right? And if you're interested in the whole cybernetical philosophy on why this is necessary and why we need more adaptive mechanisms, I invite you to read this paper, which was published in 2015. And finally, since there is not much time, this is a really short period of time, but it's fine. I will just tell you one more implication how we have to think about the brain if we start thinking this way. If we give a chance to this hypothesis. Classically, when we think about output of neurons, then we have to organize our explanation of how these neurons combine into entire performance through layers, right? So we have to, or through different stages of processing, different areas. First area feeds information to the next area, to the next area, and so on. Ever more complex processing in the next stage. That's what we have also in machine learning, in artificial intelligence, where we have layers of deep neural networks. They're called deep because we have lots of layers. Now, if we think with this new way, where we have this transient rewiring of the network, we don't need any more of these layers. Number one, actually, it cannot work well with these layers. It works much better if we think of the entire brain and all the neurons as mostly everything working in parallel, right? And if we look the connectivity, the actual connectivity in the brain, the actual connectivity of our visual cortex and cortex in general, it's highly parallel connected. Like all the parts of the cortex get and receive inputs from subcortical areas and project back. Everyone, there's no exception, right? The connections between brain areas seem to be addition to these primary connections that go to subcortical and right? And everything is parallel, and we just paralyze the brain with this much more, right? So I will stop now because there's only a couple of minutes left. Maybe there are some questions, and I'll take.
Thank you. Actually, we're a bit late on our schedule, and if there's any questions, and it looks like there's no questions, so therefore, we like to switch to the next speaker, please. Yeah, and please. Oh, there is one question where the speaker is setting up. If Alex, please.
Yeah, thank you. I was kind of wondering whether there is a connection of this rewiring theory and computations through dynamics and neural manifolds. So I don't know. Thank you.
I don't know. I don't know. The neural dynamics, you probably would have, the time constants the same as the time constants of electrical activity of neurons, and then it wouldn't be right. So rewiring would be an alternative to neural dynamics, right? Instead of looking to explain the brain activity through complex dynamics of electrical neural activity, you have something much simpler that explains perhaps the same phenomena, but it does not require dynamic like recursive activity and things like that to maintain the information, because you simply maintain information by the very fact that these proteins stay in this state for several hundred milliseconds. And this makes it much, much